Thank you. Go for it, Claudia.
Sorry, some technical issues. Wait, why can I? Oh, can you guys see my screen?
Yes, looks good.
Okay, thank you. Hi, everyone. My name is Claudia Choi. I am an undergraduate senior at the University of Hawaii. My major is molecular and cell biology, and I'm graduating this upcoming fall of 2025. And today I'll be sharing the project I'll be working on this summer, where I explore how age impacts antibody response to COVID-19. So to start, I will quickly go over the start of my presentation, which are antibodies. So antibodies play a crucial role in the adaptive immune response, and they are produced by B cells after we expose to an antigen and help the body recognize and eliminate specific threats. They are Y-shaped proteins made of two heavy and two light chains. Each antibody has a variable region, which binds the antigen, and a constant region, which defines its class. There are 5 main antibody classes, each with a different immune role. And within the variable region, there's a small segment called CDR3, and it's formed by the joining of B, D, and J genes. And it's the most diverse part of the antibody and the main site of antigen contact. So now that we covered like the basics of antibodies and the CDR3, and we know that the CD3 is the most diverse part and it binds the antigen. The next question is, how does it become so effective? So that brings us to affinity moderation. So after activation, B cells enter a structure called germinal centers, where they go through rounds of mutation, especially in the CDR3, to improve how tightly they can bind to the antibody. So basically, the best binder are keep and expanded, and the rest are removed. And over time, this create a stronger and more specific antibodies. And since the CDR3 mutates the most, we can measure how different it is from the original germline using percentage identity. So basically, lower identity usually means more mutation and better binding. So by analyzing the CDR3 identity, gene usage, and clonal expansion, we can get a sense of how strong the antibody response is. So before getting into my research questions, I'd like to share what led me to pursue this antibody-based project. So for the past 2 years, I've been part of the IMBRI program at KCC, where I've been involved in antibody research, mainly working on sequencing monoclonal antibodies. And part of that work include post-analysis of antibody sequences using various bioinformatic tools. And based on that, the data I generated myself, which can be often be like very small and like limited to very few samples. So basically that experience sparked my curiosity to go further and explore like a larger antibody data set to better understand like the immune responses on a broader scale. So as I begin reading, one study that stood out the most was a study that focused on yellow fever vaccination. It compared antibody repertoires between young and middle-aged adults and found that B-cell diversity decreases with age, with a noticeable changes already by the age of 50. So that raised an important question: if aging affects how diverse and effective antibody responses are, maybe it's something we should be paying more attention to, especially since it can impact how we approach vaccine design and immune health in older populations. So with that in mind, I decided to focus on COVID-19 since it's one of the most studied diseases and a lot of data is available. So my research question became, does age impact antibody responses to COVID-19? And based on the literature, I formed this hypothesis, which is individuals under the age of 50 will have a greater B gene diversity than individuals over the age of 50. So to achieve this, I started by downloading antibody repertoire data from iReceptor. iReceptor is a platform that hosts B cell and T cell receptor sequences data from multiple studies around the world. So I apply filters for age, species, and COVID as a diagnosis to narrow it down. And in total, I put out 13 independent studies, which cover 166 patients and had 719 distinct BCR repertoires. Altogether, this gave me around 70 million sequences, which was about 65 gigabytes of data, and all of this was in TSV format. I then split the data into two groups, under 50 and over 50. Basically, the main reason for me choosing this cutoff was because it offered the most balanced comparison in terms of number of patients in age group. So here is the general workflow I follow. So first, I access the public BCR datasets through iReceptor and inspected the metadata to assign each repertoire to either under or over 50 group. I then download the TSV format, the data in the TSV format. I next, using Python scripts, I process the data by splitting the repertoires into under and over 50 age groups. I filter the sequences that were non-productive, and then I normalize the data by randomly sampling 500 sequences per group. From there, I extracted two key factors, the identity percentage and the B gene usage. One I used to measure mutation levels, and the other to explore diversity. Finally, also using Python, I ran several type of analysis, like a heat map. I ran some statistical tests. I measure identity percentage, and I created a circle packing plots to visualize B gene usage. So the first tests I ran were on the B gene diversity, looking at how many B genes were used in each age group. Using Python, I ran a few statistical tests, like the Shapiro-Wilk, which confirmed both groups have a normal distribution. Then was the Levine test, which show similar variances. So then I used Welch test, which found significantly higher B gene diversity in the over 50 group. And I also ran the Mann-Whitney U test to double check without assuming normality, and it confirmed the difference. So overall, this suggests that older individuals show higher B gene diversity, meaning that their antibody response use a wider variety of genes. So then on this side, also using Python, I created a heat map that displayed the top 40 most expressed genes among both groups, with darker color representing higher counts. And we can see that the over 50 group have a higher levels of expression in certain genes, which kind of support our statistical findings. So next, I look at B gene identity, which shows how similar each sequence it is to its original germline. With lower identity means more mutation, which usually leads to better binding through affinity maturation. So in this, we can see the blue. It's for the over 50 group, and the orange is for the under 50 group. So the younger group peaks near 100%, while the older group peaks near 95%. And this difference was statistically significant by the p-value, which was 0.0001, showing that older individuals have more mutation and affinity maturation than younger ones. Next, also using Python, I ran Bcoy and generated this circle packing plot to visualize the top 200 B cell clones in each age group. Here are some of the package I use. Each circle represents a clone. Basically, each circle represents a clone, its size and the color represent how many B cells share the same antibody. And you will notice that the younger group has more large dark circles. And one example is clone 1315, which has 5,475 B cells, making it the largest clone across both groups. This point to more clonal expansion in younger individuals. So basically, clonal expansion means the immune system identified a helpful B cell and rapidly copy it to fight a virus. So the more clonal expands, the more dominant that antibody becomes, and often reflects like a stronger and more targeted immune response. So this table also came from BCLUD, and it shows the top 10 most spending clones in each age groups with their respected BD and J identity scores. So once again, we can take a closer look to clone 1315, and we can see, we know that that was the most expended of overall, and we can see that their identity percentage was 100%. So that means that this clone was completely unmutated, and that could suggest that the immune system selected it early, and then it's selected because it was effective and it was a high affinity clone without needing much further mutation. So overall, younger individuals have higher identity, which means less mutation, and older individuals have lower identity, which suggests more affinity maturation. Yeah. So while younger individuals show more expansion, older individuals produce more refined mutated antibodies, which it's kind of interesting because it highlights the difference between immune strategies depending on the age. And then, so to wrap things up, there were a few limitations. I couldn't fully normalize the sequencing count. There were more people in the over 50 group, and some individuals have multiple samples. I also didn't separate the data by disease severity or vaccine status, and more detailed age breakdown might have revealed additional insights. Even saw some key findings. I found some key findings that stood out. Like, I originally expected younger individuals to have higher B gene diversity, but the data show the opposite. Basically, other individuals have more. Like, this may suggest a stronger antibody response in older adults, possibly due to infection, vaccination, or other factors. For future work, uh, I would like, I'd be interested in exploring more about the clonality. And on a personal note, I'm still building confidence in coding and data analysis. I'm still having a hard time with the statistical tests, so I'm hoping to get better at that. And just also, I'm hoping that I will learn more about how to explain this biological concepts better. So yeah, I'm excited to keep learning. And here are my acknowledgements. Thank you to all the mentors. Everybody was very helpful and kind. And also thank you to Dr. Sean Cleveland for putting this grant together. And I wanted to say a special thanks to Dr. Brian Hunt, Massive TC, KCC, because he gave me a lot of good ideas on how to present this data. And here are the references. Oh, I don't know. Sorry. Oh, and then thank you.
Awesome. It's a very complicated set of data you're analyzing there.
Yeah, it was really hard for me to explain everything and like just make sense of everything.
Yeah. Wow.
I think
Does
you did a
anyone
great
have a
job, though. I just wanted to say, like, I just want to encourage you that it is a intrinsically challenging concept just to get your head around, let alone to work on. So, just want to keep encouraging you that you're doing the right thing. Also, like in terms of your statistics and you're looking for significance and normality and also significant differences. Like that's how science is done, and it'll get easier with more practice. So just keep at it. And yeah, really informing us as the audience of why it matters. And I think you did a great job doing that.
You.
Okay. Yeah, I thought this was a great talk. I saw that you were using iReceptor in some of your slides. So what did you think of iReceptor?
At first, I had such a hard time like finding the data I wanted to use because I was focusing on age, and I guess a lot of studies do not display that information. And I don't know, I just have such a hard time finding the data to begin with, but maybe it was just me. I don't know, it wasn't as intuitive as I thought it would be.
Okay.
Okay. Thank you very much. Very nice talk there.